Hi, everybody. Hope everybody's doing well, staying safe. For the younger folks out there, one of the things that you have to do periodically in science and in academia is you have to give talks about your life's work. So I recently had to do that, and I didn't really want to talk about my whole life's work. But I wanted to focus on things that we've done more recently, i.e. in the past decade, which is mostly mannequin things. So the talk that I gave was mostly about mannequins, but I thought I could expand it a little bit for this group, sort of capture some of the history of what we've been doing on golden-collared manakins. But just so you have, for those of you who don't know me, my background was, not only in ornithology and bird watching and all, but I was very interested in hormonal control of behavior, the hormonal actions in the brain, and that background led me into how I got started working in mannequins, as you'll see. So let's see. I'm trying to move it forward, and it's not going. And I had this problem once before. It worked just a minute ago. There you go. Okay, so I think the main thing to point out is that a lot of the work that we've done involves many of the people that you know. I'm going to speak quite a bit about work that Joanna Cheever has done recently, but there's quite a bit of work from Matt Fuchsjaeger, from Leo Fusani, Virginie did a little bit of some things involved with mannequins. Lainey Day, Jenny Feng was an undergrad in my lab who has participated in some work. Some of this work started early on with a guy named Fritz Hertel, who, he was just a friend of mine who we traveled in Panama and did some work together. I had an early graduate student named Doug Schultz. We did some work with Ted Goslow, doing some early work with high-speed video. Then I had a number of collaborators, Martin Wikelski, Ayla Howe and Manfred Gar's group at the Max Planck, Dan Bedoni at University of Illinois, Grace Xiao and Tony Frisia at UCLA, and Franz Gohler at University of Utah. And all this work's been done through the Smithsonian Tropical Research Institute, and I've been funded by NSF for a number of years to do this, and a little bit of money from NIH and American Heart Association in UCLA. All right, now why is it not going? There it is. Okay, so again, just by way of background, a lot of work that we know about where people have studied neural and endocrine basis of behavior has looked at sex differences and hormonal control, but is largely focused on very highly stereotyped sex behaviors like these in rats, where the female shows her stereotyped lordosis behavior and males do his stereotype set of mounting behaviors. And many of the systems that have been explored have been relatively simple, like the muscles that control the penis in the male rat, this SNB system, where we know about hormonal control of the neural systems as well as the muscles. But as we all know, many highly complex behaviors can be sexually dimorphic or hormone dependent. A good example is the work that's been done on the Oscine songbirds, where the males sing to females. In the zebrafinch, the circuitry that you see on the right side here encompasses the auditory processing system, the motor output, centers that are involved in learning, and the discrete control of the motor control of the discrete muscles in the syrinx, illustrated here. So there's this very complex behavior that is basically, results from this very fine control of this tiny little set of muscles in the syrinx. And I was very interested in the golden-collared manakin. I had traveled to Panama bird watching and had been at STRI and been at Bower, Colorado, and met people, and I saw golden-collared manakins doing their display, and I thought, wow, these are exceptionally complex behaviors, and can we learn something about the neuroendocrine control of that behavior, and how does it differ from other kinds of behaviors? And I started out doing some research and was struck by the fact that Frank Chapman back in 1935 had published a really terrific monograph from his work on Bower, Colorado, that described a great deal about Manakin's vitellitis that used to be called Gould's Manakin. So we had a good background of behavior and a good logistical place to work in the Smithsonian. My first attempts to study these birds done with Fritz Hertel and my first graduate student in this area, Doug Schultz, involved where we traveled to Panama, injected birds with radioactive testosterone and looked at spinal cord accumulation of radioactivity, presumably because, I mean, with the idea that there are many muscles involved in the Manakin courtship and there must be tremendous accumulation and action of androgens in the spinal cord. This is the image on the left is just showing you the distribution of these androgen concentrating cells in the spinal cord. We found sex differences with more in males than there were in females. And these areas where there was high concentration were in these enlargements in the spinal cord where the motor neurons that control the leg and wing muscles are located. So this was exciting for us in the early stages. We thought if we looked in the brain that we might find some very interesting brain regions that controlled production of their behavior. We're quite disappointed. The middle panel shows some staining in a songbird, and you can see that there's this area X. You can see this dark stained area here of this nucleus HVC, this nucleus RA. And if you look in the area of the Manakin, the same brain regions, you see nothing that stains either with a specific antibody that was found to stain the neurons in the song system, but also with just a general stain that stains neurons in the brain. Nothing really stood out. Clearly the brains of the Manakin were different than the songbird, and Laney did some additional studies a number of years later that showed some interesting aspects of the visual system, in fact, which was quite dominant. But anyway, this was some of our earliest work. But all along, we were really struck by this fact that there were these extraordinary sex differences in behavior and phenotype. Of course, we were very interested in theories of sexual selection that would lead to the production of these very complex, unusual behaviors, and this was part of what drove our research early on. And so we really began thinking of the idea that the female Manakins were choosing males based on their superior physiological function. We all know that Manakins are very long-lived birds. And many of these males who are doing these very complex displays live a long time. You know, Dave McDonald, if he ever made it, if he's out there, you know, his long-tailed Manakins that are, you know, live to be 19 years of age is really quite extraordinary for a 25-gram bird. But we were also, you know, predicting that there were discrete neuromuscular specializations that females might be choosing, using this displays as a mechanism of choice. And in later years, we began to appreciate the fact that there may be really optimal metabolic capabilities in these birds. And it's all revealed to the females by their extraordinary courtship displays. And we took a two-pronged approach, really, one of which was to, you know, examine the male's physiological specializations, in particular looking at endocrine and neuromuscular specializations, but also asking questions about what females were choosing. For those of you who don't know the Golden-collared Manakin display, it's got visual and acoustic features to it. The males clear a courtship arena in the forest around small saplings, and they attract females that will come and join them in a dance. They emit a number of different vocalizations. These chipus are the primary reproductive vocalization, and they have produced these powerful wing and roll snaps that, and those of you who know all the Manakin species, they produce something very similar to all of these. So this is just a quick video. You'll hear some roll snaps in the background. They're males, you know, they gather in leks, and anywhere from 5 to 20 males, and they each have their 1 or 2 of these courtship arenas, and they perform this complex display. So Leo Fusani pretty much took the lead early on with some of the high-speed video work, and we had some very entertaining, entry into this whole work, because we started working with a fellow named Ted Goslow, who was at Brown University, who had a $70,000 camera system that could record high-speed video, that we schlepped it all down to Panama with, I mean, literally hundreds of pounds of batteries and the camera and computer equipment and all. And we were out in the field, and when it started raining, we had to pack it all up and run away, and it wasn't waterproof. And then of course, it took us a long time to get any of this stuff going. And then the next thing we knew is that, Rick Prum and Kim Bostwick had just purchased a small, camera that was portable and waterproof and easy to use, and they got a lot of data. So so we abandoned our dinosaur equipment and bought a high-speed video. But Leo spent a lot of time, and Julie Barsky, you'll see in a minute, collected a lot of data from individual males. The point of all this was to just point out that what Leo found was that individual males are different in their behavioral performance, and that can be recorded to the level of millisecond differences in behaviors. And you can see this with the beard up behavior, where there are significant differences. The slowest male took 62 milliseconds, and this, fastest male was 41 milliseconds. And other behaviors, the time they spent on perch, the jump speed, the distance of the jump and all, there were significant differences between individual males, which suggested a basis for female choice. And later on, Julie Barsky, who was a master's student with Leo, who then came to my lab to get her PhD, did even more videotaping of males in the field, and found that females were gaining more copulations, or were being joined by females more. And the males that got more of these copulations, or were joined by females, they performed more displays, they called more, they performed more roll snaps, and they performed more wing snaps. So the athleticism of the display seemed to be related to how much copulation they were able to gain. And there was an inverse correlation with the speed. So the faster the males were at getting this on perch display, or for getting their beard up, the faster they were, again, more copulations. So speed and the intensity of the courtship seem to be very important parameters for choice by females. The fact that these wing and roll snaps were so interesting suggested that there were probably skeletal muscle specializations. And this had been shown initially by this fellow Percy Lowe, who received from carcasses from Frank Chapman, published a paper back in 1943, showing that males had these enlarged gluteals and this large muscle on the back, which we now call the scapulohumerus caudalis. And so we were quite interested in exploring these muscles more fully. I did an early set of experiments with my PhD student, Doug Schultz, where we were able to stain several muscles, including the scapulohumerus caudalis and the supracoracoideus. the wing the muscle that lowers that lifts the bird's wings. You'll see more later on with the pectoralis, the muscle that lowers the bird wing. But you can see that there were sex differences in the staining for this fast myosin. And there were some other more subtle sex differences in fiber size and all. The males produce these wing snaps, the females generally do not. So that seemed to be a basis for some of the difference in the sex differences in behavioral performance. But we were, again, even though Bostwick and Prum had published showing that the males are lifting the wings over their back and throwing their wings together, we were still quite curious about the actual mechanisms involved in producing the sound. This was some high-speed video taken by Laney in captivity at 2,000 frames per second that was synced with audio. And you can see that the audio signal here, this is the precise moment when the snap is produced. All of this signal here is echo. But you can see that the bird is lifting his wings. This is there's a half a millisecond between each of these frames. And it's right at this point where the wrists strike each other that the sound is being produced. And we did some modeling of this behavior that indicated, in fact, that the only likely mechanism to produce the loud snapping sound was because of the bones striking each other and their instantaneous vibration that would produce that sound. It didn't seem to relate at all to the movement of the feathers or any other ideas that could be imagined. So we published this paper with Dan Bedoni, who is this aeroacoustic engineer at University of Illinois. But we followed up on this with Matt Fuchsjaeger and Tony Frisia at UCLA, where we did microCT analysis. Tony did microCT analysis of the manakin bones. And you can see that at top, the golden-collared manakin, then the white-collared manakin, and then the red-capped manakin. The red-capped manakin has a very more typical looking radius, which is elongate, rounded, but the golden-collared and the white-collared, the Manicus species, have these highly flattened radii with these enlarged both distal and medial ends. And furthermore, that the radius is mostly solid, not hollow like many of the other birds, including the radius of other manakins. So there were these skeletal specializations that aligned with our idea that indeed the bones were striking each other and producing this loud vibration sound. We were also interested specifically in the muscles. We'd done some work on them before, but nobody really ever demonstrated which muscles were involved in the wing snapping, although we certainly suspected which ones were involved. But we were able to do this EMG recordings using radio telemetry from some captive manakins. Matt Fuchsjaeger did this in collaboration with Franz Gohler, and we got some radio telemetry equipment from Manfred Gar's group at the Max Planck. What you can see here is that here's a sound recording. If you can see my little arrow, you can see here's the instantaneous moment when the snap is produced. You can see it again over here. Here's the snap being produced, followed by some echoes. The pink phase is the period of time when the wing is being elevated, presumably by contraction of the supracoracoideus. And this blue phase involves the wing retraction, where the wing is twisted so that the tips of the radii strike each other. And you can see that that's exactly how these muscles are contracting. You see the SC muscle begins showing increased contraction strength and frequency in the pink phase. It's illustrated here and here. And that right at the moment when the wings strike each other is when that SH muscle shows this profound increase in contraction strength and frequency. And you can see that here as well. And the pectoralis doesn't lower the wing until 100 or so milliseconds later. So there's this discrete sequence of events where the muscles are firing at discrete phases to produce this profound snap. But it's this back SH muscle which seems to be most important. And as you may know, Matt has, when he started his own lab, has done quite a bit of work together with Franz to show that that particular muscle has other adaptations to produce these very fast contractions. So back to looking at hormones. So our lab and Martin Wikelski's lab early on had looked at, and this was done with Laney and Leo early on, where we collected blood samples from birds throughout the seasons and found that indeed testosterone levels are elevated at the beginning of the dry season when their courtship behavior starts. And it lasts, somewhat elevated throughout the entire period of their courtship, which can last 6 months or more until courtship behavior declines. And it's during that entire 6 or 8 month period when males are at or near their arenas and pretty much daily performing courtship. By contrast, the females and juvenile males have low testosterone during that period of time, and in many cases basal and undetectable. And for those of you who don't know, these juvenile males and females are morphologically quite similar. Juvenile males have this all olive green plumage like the females. In the field, it's extremely difficult to study them, and you can't, it's hard, when birds are flitting around quite rapidly, it's hard to say for sure, is that a male or is that a female, or is that a juvenile male or is that a female? So to make any really comprehensive studies of the green birds, we really needed to either capture them and band them so that we could be sure what they were, or we needed to find a different way to study them. And the way that we did that was, we were quite lucky that we were able to acquire use of this aviary that was down Pipeline Road by this Discovery Center, and Mike Braun helped us and others helped us to gain access, and we were able to cover it with mesh that kept the mannequins in it. And that's Matt inside the aviary, and this is Matt still inside the aviary. You see when we inherited it, it was empty. But we trucked in dirt, and this was done with Joanna and Matt. Mike helped, and especially people at Stry were really helpful. And we trucked in dirt. We acquired some plants from Stry. We planted them. And, the great thing about a rainforest is that even if you don't have a green thumb, everything grows, like it or not. And everything grew, and we were able to produce a functional lek inside this aviary. And again, I just wanted to have an advertisement here for people that we did build aviaries in Panama, and I don't think they're being used now. I think there were some problems with the mesh, and I don't know if that's been repaired, but please, they're there. They're right near the facilities for doing lab facilities, so please feel free to use them. They're just begging for use. Okay, so Joanna Cheever took the lead on these experiments, and what we did was we collected birds that were the green birds. They were genetically sexed. Then they were given testosterone, and a week later, after 1 week, they were then released into the aviaries in same-sex groups. They were either 3 to 4 birds per group. They were either all males, green males, or all females. They all had low levels of testosterone, and they were either treated with, as I said, for a week with either testosterone-filled implants or blank implants. Then they were released in the aviary, and Joanna recorded their behavior daily for 3 weeks. And I just want to show you this video of their behavior. This is in fact a juvenile male. They amazingly, quite quickly can perform roll snaps they quickly will claim a court. That little display where they jump to the ground and they fly back up with that grunt is this display that they use just before they copulate with females. They do fight over these courts. So these are all three males are interacting at the court. That's a fight between two males.
Hey, Barney.
Yeah.
Are there you know, birds that were blank implanted in the same experiment, or they all have T or don't have T?
No, they all either had T or they all didn't have T.
Okay.
Yeah. This male is pecking at the ground, and this is part of their behavior where they clean their courts. We call it gardening behavior. And this is a male. We would throw dead leaves or leaves in their display courts, and they clean their display courts by removing leaves. So, so they basically do everything they would do normally in the wild under these semi-captive conditions. So when we analyze the data, and I'm not going to show it all here, but if you just look along the bottom panel with the red arrows pointing, there's either females that were treated with blank implants, females treated with testosterone, males treated with blank implants, or males treated with testosterone. You can see here that females with or without testosterone never performed any of the gardening behaviors. They never tried to claim a court. The males, clearly testosterone amplified their motivation to clean their courts of leaves. Again, females with blank implants did no roll snaps, but indeed females with testosterone could perform roll snaps. We'd seen females with testosterone in captivity in small cages perform wing snaps, and so we knew they could do it, but it was very interesting to see that they really could do it in the wild under these conditions as well. Notice this is one female here who did a lot. Even without testosterone, with low levels of low levels in blood, the males did perform some roll snaps, but again, amplified quite a bit by testosterone. So I'm not showing you all the different behaviors, but there were some just very interesting sex differences and things. And I'm going to talk more about a general perspective on sex differences in mannequins when I give my talk for the SICB. So I hope it's only a 15-minute talk, so but I hope that you can join and see some of that. So one of the other things that we were, again, interested in is where androgens were acting to control behavior. This is some work that we started back with Doug Schultz back in the early 90s and continued on and eventually published it in 2013 with Matt Fuchsjäger, but showing that there are spinal motor neurons and their sensory neurons in the dorsal root ganglia. These are backfilled by dyes that were injected into either into the SH muscle. In this case, it was this fast blue, and in this case, it was fluoro gold. And then we did what's called in situ hybridization to look for expression of androgen receptors. And you can see that here, when you overlay these, you can see that the motor neurons and the sensory neurons that innervate these muscles express lots of androgen receptor. And Leo did very similar kinds of experiments in the lab on brain areas. And again, this was work that he did back in 2002 or so, and took us a few years to get it published. But there were some interesting areas of showing high concentration of androgen receptors. Here is this archipallium region, which has premotor neurons that control, it controls song behavior, but may also control the neural circuits that are the basis of this courtship behavior. There's a lot of androgen receptor in the cerebellum, which we know is involved in motor control, as well as some mid and hindbrain areas as well. But it seemed like that the androgen receptors that are expressed in muscle were especially important. And this was, I'm going to talk about it in a couple of different ways. So this is a male doing his roll snap. That entire sequence, I'll play it again, is about 300 milliseconds long. So a third of a second to do all of this. And you can visualize, something like that here. This is a schematic showing these individual snaps over the course, in this case, about 250 milliseconds. This is 1 second, this whole duration here. And there are both these individual snaps, but then there's the gap, the interval between the snaps. And I'll talk about that in just a second. But one of the things that Matt did in my lab was to go into the field and to treat birds with a drug that was a selective androgen receptor antagonist for peripheral androgen receptors. It didn't supposedly cross the blood-brain barrier. Leo had done some work a decade earlier with a drug called flutamide, which also reduced the behavior of birds, but flutamide, it was a bit messy, and flutamide crosses the blood-brain barrier, but we were interested in these androgen receptors in muscle. And sure enough, what Matt found is that these were blank implanted birds. You can see they do a lot of wing snaps and roll snaps and the whole jump snap display. But the birds that were treated with bicalutamide showed very little, if any, behavior whatsoever. So blockage of these peripheral androgen receptors seem to be really have a profound effect on reducing the performance of these particular behaviors. And chipus, by contrast, there was no difference. There was a little reduction, but it wasn't significant. But if we go back and look at these roll snaps, again, it's just showing you the schematic again. If you look along the bottom, what is plotted here is the interval between snaps. So this one male, many of these males who are treated with blanks, as you can see the dark line here, there's about a 20 millisecond interval between the wing snaps. And males could do 15, 17, 19 of these wing snaps in a row. One did up to 24. So there's 23 intervals. So that's one impressive thing. The other thing that's impressive in my mind is that if you look at this line, it's relatively flat and the variance around each of these is relatively small. So this 20 millisecond time interval is very, very tightly controlled for long periods of time. And the longer the roll snap goes, it seems to get a little bit more variable. But if you treat the birds with this androgen receptor blocker, they could only do as many as 7 or 8 of these wing snaps. And the interval was slower and the variance was quite a bit higher. So androgen receptor expression in the muscle seems to have very tight control over these fine motor skills that these males are performing. And again, we now wonder if females are able to judge the length of these displays and the tightness of this, or the skill or the fine motor control of the displays, and that remains to be done. But we were also quite interested in the fact that when we looked at androgen receptor expression in Manakin skeletal muscles, that the expression levels were very high relative to other birds. And this was done by Jenny Fing, who was an undergrad in my lab, doing quantitative PCR. And you can see the Manakin expression in all of the muscles, the supracoracoideus, the pectoralis, the scapulohumerales caudalis, the gluteal and the syrinx. There's all much higher in the manakin than in the zebrafinch or in a ochre-bellied flycatcher. And what was also interesting is that in the manakin muscle, skeletal muscle, it was higher than in other regions of the manakin. It was higher expression than in brain, spinal cord, or even in testis. This wasn't significant, but it certainly looks to be higher. So they really, this exceptionally high expression of androgen receptor in their muscles seems to be a trait of importance. Matt, when he was in my lab, tested this by examining different manakins and non-manakin species together, looking at androgen receptor expression across in muscle, and looking at birds that differed according to their display complexity. And this was work that was based on display complexities that were published by Rick Prum previously, and then modified by Laney and her postdoc Willow, and I can't remember her last name, but she doesn't really need a last name. But again, these were birds that were ranked according to their display complexity, with the zebrafinch being quite low. They have very minimal display. Pin-tailed whitas that were collected from Puerto Rico, they have a bit more of a display with these long tails that they have. Ochre-bellied flycatchers have a bit of a wing display. And then several manakins, that again, the golden-collared had the, by these measures, the most elaborate courtship, the blue-crowned manakin a bit less so. But when we looked at androgen receptor expression in muscle, there was a very tight correlation between the androgen receptor expression in their wing muscles relative to their courtship displays, whether we looked at their total display complexity or just those parts of the display that involved wing movements. But these adjusted R-squares of 0.76 and 0.71 really suggest that androgen receptor expression is a very powerful predictor of how elaborate the courtship display or their muscle use is going to be. So the results provide evidence that selection acts specifically on androgen receptor expression to guide these phenotypes. We published this in Functional Ecology, and we were pleased that there was a spotlight article that was written that discussed this further and was really pointing out that androgen receptor expression really could contribute to the honesty of a sexual signal. They can't fake it, and be this basis of these species differences in courtship. So I'm going to just, I know I'm going to kind of going a little fast. I'm going to just summarize briefly where we're at. It seems that females choose males based on some kind of display performance, which, I've used the word superior in quotes. They seem to be more intense and faster. There are anatomical, both skeletal and muscular specializations, and androgens play a really prominent role in these behavioral performance. I'm going to talk about a little bit about this integrated behavioral phenotype. There's the physiology group of the RCN has a paper that is still being produced, but it discusses this integrated behavioral phenotype, that maybe there are other physiological systems that are adapted for display performance and that are also sensitive to androgen control. So I want to just point out, if you look on the right side here, Julie Barsky did some work where she put audio recorders out to just record birds' activity across the day. We pretty much knew that they were more active during the morning and the evening, and with little activity during the day. And this just illustrates this for a group of birds, showing higher activity in the morning and the afternoon, and a lower activity throughout the middle of the day. In some days, they're completely silent during these midday periods of time. But we were also using we used radio telemetry to get heart rates of birds. Again, this was in collaboration with Mark Mikulski, and several of us, worked on this, and including a group of Princeton students in Panama who were willing to stay up all night and we're happy to do so and to help us collect these radio telemetry recordings of heart rate. And you can see that the heart rate is a little bit lower at night. You just follow this mean bar, the dark bar. It rises precipitously in the early morning at a time when they're waking up and they're performing courtship displays. It lowers in the middle of the day and it rises again a little bit later in the afternoon before it declines precipitously again at night. And we don't know whether this is just sleep or represents some kind of torpor, but it's a pretty dramatic change in heart rate across the day. And in particular, when the birds were doing their particular courtship displays is when we saw some really exciting things. That when the birds were making roll snaps or when they were in the middle of their jump snaps displays, the heart rate really accelerated profoundly and rapidly to up to over 1,000 beats per minute. It only did it for 10 or 15 seconds, but it still was very high for a short period of time. And then when the birds were just at baseline or doing other behaviors, it was around 550 beats per minute. So there were these really rapid changes. Moreover, we were able to predict their daily energy expenditures because we took birds into captivity and were able to use calorimetry to determine their oxygen usage and carbon dioxide output and relate that to these heart rates that we obtained. This had already been done by Martin Lukelski in spotted antbirds. And basically, I've been going quickly here, if you just look down here at the bottom, that even though manakins are performing these very powerful displays and doing many, hundreds of such displays throughout the day, in the morning and the evening mostly, that their overall daily energy expenditure was no different than that from a spotted antbird. And I know Virginie or others may have their opinions about spotted antbirds, but to me, they're, you see them in the forest, they give this very soft, sound, song. They move about relatively slowly. They don't seem to be a robustly vigorous, active bird like the Golden-collared Manakin. Yet they still use about the same energy per day in their lives. So does that suggest that there might be cardiovascular or metabolic specializations that the manakins have evolved to enable them to do their courtship displays? So one of the things we looked at at hearts of some manakins that we had collected. There was some previous work from a fellow back in 1955 who just weighed hearts of all sorts of different kind of birds and showed that manakins had slightly heavier hearts. And we were able to show that when we compared, this was again, Julie Barsky did this in my lab, that when she compared the Manakin heart here compared to a zebrafinch, they're much thickened in the areas that involved in contractions of their left ventricles and in the septum between the left and right ventricles compared to the zebrafinch, suggesting that in fact this larger size and this larger weight is based on this more extensive myocardium that the Manakin has. But another thing that stood out quite strikingly is we looked at a number of genes that were expressed in the heart, and the Manakin heart has much higher androgen receptor expression than does in the zebrafinch, suggesting that there could be some androgen dependent genes. We looked at a few possible candidate genes and found one that was regulated by androgens that could relate to the speed of the contraction of the heart, but a much larger study is actually needed to explore this fully. But there is a possibility that the manakins possess cardiovascular specializations that enable them to perform their courtship displays. We had also done work looking at collected muscles and did RNA-seq analysis on the transcriptomes of muscles from manakins and zebrafinches. And in this case, we looked at the scapulohumerales and the pectoralis. I don't really have time to go into great detail here. There were differences. The pectoralis was quite different than the scapulohumerales in both the manakin and the zebrafinch. But androgens had a much greater effect on, as we had expected, had a much greater effect on the manakin muscles, in both muscles than they did in the zebrafinch. But one of the things that stood out when we looked at those genes that did seem to be regulated by androgen, we kind of expected that we'd find genes related to um, contraction rate or ion transport. But we did notice, and just pointing down here, that there were a number of genes that were involved in metabolic processing, including lipid and glycogen metabolism. Again, suggesting that androgens might have a specific effect on the muscles to improve their capacity to use whatever energy is available in allowing them to perform their elaborate courtship displays. So, so metabolism, cardiovascular specialization, the display performance that you witness, there are all sorts of things that seem to be able to contribute to them performing it. As I say, metabolic, cardiovascular, the neuromuscular, physiology, and androgens have the capacity to act on all of these systems to create this integrated behavioral phenotype that involves all of these different organ systems and physiological systems. You know, add on top of this their longevity, which includes that can be practicing more their behavior from year to year to year, that might contribute to some of these specializations, but also contributes to the optimality of the display performance. The display becomes a remarkable reflection of their anatomy, their physiology, and their longevity. So again, I'm just shouting out a shout out here for this paper that hopefully will be coming out some year by the physiology group, where we can really look at all of these different systems. We haven't talked about the sensory systems. Clearly, females have the capacity to view the very high speed of these displays, and Laney collected some evidence that the visual systems of the females are enlarged compared to the males. There's a lot of work that needs to be done on this. I know I know Leo and others have tried to do some work in this area. I tried and I failed totally. But these are just things that I hope others are working on. And so again, when we think about sexual phenotypes, and there's all sorts of theories out there, you know, there are all sorts, you know, all of the theories for how females are choosing males are highly contested. Mannequins fit squarely within this whole realm. And, you know, Rick has obviously done a lot of work in this area. And I just want to end by pointing out that I don't forget that these physiological and anatomical systems driven by direct genetic control or by hormonal control can have a powerful effect on features that are chosen by females. And that kind of describes a lot of stuff that's been going on in my lab and related labs on the Golden-collared Manakin. And thank you all for being here, and I'm happy to answer any questions. And I hope I've done credit to all the people that, you know, worked on this. You know, like I said, Leo and Julie and Laney and Matt and Ioana, and I've been really, really lucky to have worked with a lot of really fabulous scientists and people, and I'm glad I get to still see them all the time, even if just by Zoom. So there you go.